Every year, 6 million North American dogs are diagnosed with cancer. 1 in 2 dogs over the age of 10 years will receive such a diagnosis in its lifetime. Cancer represents hundreds of distinct diseases, and more treatments are required to improve survival rates and improve the quality of life of our patient animals. Canine oral melanoma has a median survival rate of only 1 to 6 months for stage 2 to 4 tumors, despite aggressive surgical excision. Compared to other types of cancer, melanoma causes an immune response in the body, which means that it's amenable to something called cancer immunotherapy. Immunotherapy is a form of treatment whereby the immune system is strengthened or restored to fight cancer. In 2013, immunotherapy was named the Breakthrough Discovery of the Year by the journal Science. Now, one option to treat melanoma is to administer a virus called AAV that carries a gene inside of it. Once inside the animal, this gene is turned into a protein, essentially turning your dog into a protein-making factory with the cure. Now, hundreds of studies have shown that gene therapy is a safe and effective method to administer treatments. And they have also been used to successfully treat a wide range of illnesses, including cystic fibrosis, hereditary blindness, and hemophilia. Now, gene therapy is especially useful for cases where the drug is too expensive to create in the lab, it's very difficult to make, or finally, if it requires constant clinical visits. For my project, I'm aiming to use AAV that delivers a protein that recognizes something called PD-L1. PD-L1 is a protein that sits on the surface of our immune cells called a T cell. And this protein regulates the immune system. When there's more of it, it reduces the number of T cells, which in turn makes it harder to kill the cancer. Now, cancer has found a way to take control of PD-L1 to avoid detection. In doing so, the cancer has essentially made a deal with the devil, and melanoma is about to find out that it does not like the terms of the contract. For my thesis, I'm creating an AAV that delivers a gene that has something called an inducible promoter. Now, with this promoter, we can turn it on or off, which is important because it reduces side effects for the animals. I aim to turn long hours in the lab into long walks in the park for other labs. I'm going to put forth a woman's best effort to find the cure for man's best friend. Thank you.